Welcome everyone to today's Connect Science seminar. It's a pleasure to introduce Cynthia Wohlberger for her talk. Cynthia is a professor of biophysics and biological chemistry at Johns Hopkins University School of Medicine and is a world leader in the mechanisms and structural biology of ubiquitin signaling and transcription and the DNA damage response. A little about Cynthia's background. After obtaining a BS in physics at Cornell, Cynthia performed her PhD in biophysics in the laboratory of Steve Harrison at Harvard University, where she was co-mentored by Mark Potashny and carried out a series of studies to understand how bacteriophage DNA binding proteins specifically associate with particular DNA sequences. She then performed her postdoc in the lab of Carl Pabo, first at Berkeley and then at Johns Hopkins. There she began her work on understanding how protein DNA interactions at the mating type locus in budding yeast. Since starting her own lab at Johns Hopkins, Cynthia's made groundbreaking discoveries across several fields. Her work is characterized by beautiful experimental design coupled with biochemical and structural clarity that provides a rich landscape for mechanistic understanding of complex biological systems. After building her early career on homeodomain DNA binding proteins, Cynthia became interested in regulation of chromatin through multiple mechanisms. But perhaps the most impactful work is her elucidation of the structural mechanisms of the sirtuin family of NAD-dependent deacetylases, which led to an interest in nucleosomes and the histone code. In parallel with work on sirtuins, Cynthia initiated her work on the ubiquitin system together with the late Cecile Picard, providing the molecular mechanisms by which ubiquitin conjugating enzymes promote linkage-specific chain assembly. This included K63-linked ubiquitin, which plays a central role in DNA repair. This interest in the ubiquitin system then coalesced with her work on histones and transcription through analysis of deubiquitinating enzymes, such as the SAGA complex, that removes ubiquitin from histones within chromatin. In particular, her work revealed the structural basis for regulation of the 4-protein DUB module within SAGA and provided key insights into why many DUBs have associated regulatory factors that control their activities. Most recently, she has revealed how the COMPASS complex recognizes ubiquitinated histones, which was really a tour de force study. For all this work, Cynthia has received numerous honors and awards, including the election to the American Academy of Arts and Sciences and to the National Academy of Sciences. She is also heavily involved in educational activities at Johns Hopkins and is a major voice for diversity in the life sciences. And so today, Cynthia will tell us about the latest on the role of ubiquitin in regulating chromatin modification. So if you have questions, please put them in the Q&A and we'll address those questions at the end of the talk. Cynthia?
All right, well, thank you very much for that very nice introduction, Wade. And I'm sorry I can't deliver this talk in person and be there to see so many friends and colleagues and others of you that I've yet to meet. So as Wade, oh boy, already, there we go. So I'll be telling you about the work that we've been doing on the role of ubiquitin in regulating chromatin modifications. So to begin at the beginning, so our genomes are packaged into a nucleoprotein complex known as chromatin. There are the 4 core histone proteins that form this, organize this fundamental unit known as the nucleosome, and then nucleosomes in turn associate to form higher order packaging of chromatin. And if we were to compare this open organization of chromatin, so this is euchromatin, which has nucleosomes like beads on a string, and is what chromatin and actively transcribed regions are, versus heterochromatin, which is highly packaged and transcriptionally silent, you would see that the histone proteins have very different distributions of post-translational modifications. So our histones are subject to really an incredible variety of post-translational modifications of a vast chemical assortment. And it's been found by research in multiple fields that histone modifications of a particular type at a particular residue have specific roles in many cases in regulating transcription, as well as pretty much everything else that has to do with accessing our DNA. So the modifications vary enormously, not just in chemical properties, but in size. So there are smaller chemical modifications like acetylation, methylation, phosphorylation, succinylation. And you see an example here of a methylated lysine. So here's a CPK model of lysine. There's the methyl group. And all the way up to very large modifications like ubiquitin. So this is the ubiquitin protein, the 76 amino acid protein that is covalently attached to lysines via its C terminus. And this is the modification I'll be focusing on and the role of this modification in regulating methylation. So the role of ubiquitin I'll be talking about is not its very well characterized role in targeting proteins for degradation by the proteasome, but rather its signaling role in regulating gene expression. So this is a non-degradative role. So histone monoubiquitination, so attachment of a single ubiquitin to a lysine residue, doesn't have a particular meaning or function. It also depends where that ubiquitin is attached. So monoubiquitination of histone H2A at lysine 119 is a modification found in heterochromatin. So that's associated with transcriptional silencing. Whereas monoubiquitination of histone H2B at this specific residue, so in humans, it's lysine 120, it's 123 in yeast, plays a role in active transcription. So it's prevalent in actively transcribed genes. And I'll be focusing on its role in transcription, although I should say it really plays a variety of very interesting roles also in DNA replication, DNA repair, and in nucleosome dynamics. So we've been studying a variety of aspects of the role of H2B ubiquitination in transcription. So as Wade referred to, we've been working on the SAGA deubiquitinating module, so part of the SAGA coactivator and understanding how that specifically removes ubiquitin, as well as the very interesting role of a different H2B specific deubiquitinating enzyme, UBP10, in regulating nucleosome dynamics. But what I'll be talking about today is our work on the crosstalk between monoubiquitination of histone H2B and regulation of two different methyl marks. So if we looked across a transcribed region, so this is denoted below here. So this is RNA polymerase II poised in the nucleosome depleted region, and here's our open beads on a string chromatin. So if you look across a typical transcribed region, there'll be ubiquitination of histone H2B peaking at the start of transcription, but stretching across the entire gene. There'll also be methylation of histone H3 lysine 4, and that's particularly clustered around the promoter region. And then methylation of histone H3 lysine 79 is distributed across the gene. The connection between H2B ubiquitination and these two other marks was first discovered in yeast, when it was found that if you knocked out any of the machinery involved in attaching that ubiquitin to histone H2B, or if you mutated the lysine that it's attached to, you also lose methylation of histone H3 lysine 4. And so the missing band here is when you knock out an E2 enzyme, as well as lysine 79, another methylation mark. And these are Western blots against specifically those marks. And then it was subsequently shown by Bob Rader's lab, but the same is true in humans. So the H3K4 mark and the H3K79 mark are also both dependent on ubiquitin. And by the way, for the rest of my talk, I'm going to use this shorthand. So H3K4 means histone H3 lysine 4, H3K79 histone H3 lysine 79. So I don't have to keep saying those long things over and over. So methylation, just very quickly. So these are illustrations of methylation of lysine. So lysine can have up to 3 methyl groups attached. So that's mono, di, and trimethyl lysine. I'm not going to dwell on the differences between the two. You'll see throughout my talk, you may see mono, di, or tri. I'll dwell on just the general connection between the two without focusing on the three, but all three modifications can be deposited by the enzymes that I'm talking about. So what does the methylation do? So like many other histone modifications, it serves as a landing pad for domains that specifically recognize those modifications. And those domains are part of other complexes that play a role in activating transcription. So in the case of H3K4, this modification here in the tail of histone H3, there are a variety of complexes. So the SAGA coactivator has an H3K4 recognition domain, a nucleosome remodeler, as well as a component of the transcription initiation complex. So that's the role of this H3K4 modification. H3K79 in the core of the histone is a little bit more mysterious. We know it's found in actively transcribed genes and directly associated with active transcription, although its intriguing role has yet to be elucidated clearly as to what exactly it does. So although both of these modifications are dependent on H2B monoubiquitination, they're located in very different structural environments. So H3K79, as I showed you, is in the core of the histone. So this is the globular core of those histone fold of H3. And it's about 25 angstroms from the ubiquitin, whereas H3K4 is in this tail region of the enzyme, and it's actually located quite far from H2B ubiquitin. These two modifications are also deposited by two very different enzymes. In the case of H3K79, it's a monomeric enzyme, DOT1 or DOT1L, whereas H3K4 is deposited by this large enzyme complex called COMPAS in yeast and MLL1 in humans. So what I want to talk to you about is what we've learned about the molecular basis of a crosstalk between histone ubiquitination and these two modifications. And I'll start by telling you about H3K79. So H3K79 is methylated by human DOT1L, and it's correlated with levels of transcription. So the higher the levels of transcription from a given gene, the higher the levels of methylation, both monodi and trimethylation of H3K79. So the protein that does this DOT1L, it's actually quite a 1500 amino acid protein, but the business end is here right at the amino terminus. There's this catalytic domain extending to about the first 350 amino acids. And then there's a short DNA binding domain, and then this long region over here that associates with a variety of other partner proteins that form a complex with DOT1L in cells. Now, although we've had the structure of human DOT1L for almost 2 decades, and also the structure of the yeast DOT1 enzyme, which is very similar, it's never been clear how it even binds ubiquitin, much less is stimulated. That there had been attempts to make mutations and figure out where it binds. It doesn't have any known ubiquitin binding motifs at all, even though we know that we knew at the time this was the piece that is stimulated by H2B ubiquitination. Now, DOT1L is actually an important drug target. So there are MLL rearranged leukemias that affect the MLL complex here. So MLL is the enzymatic complex that methylates H3K4, and it contains the MLL protein and a bunch of other associated factors that form this large complex that target H3K4. In MLL rearranged leukemia, the catalytic domain, the C-terminal domain here, is replaced by a variety of fusion partners. And in about 40% of MLL rearranged leukemias, that fusion partner, that fusion segment is one of the partner proteins of DOT1L, one of those proteins that interacts with that C-terminal domain of unknown structure. And then that partner protein, the fusion protein, drags DOT1L into the complex. So you replace the methyltransferase and you now get aberrant levels of H3K79 methylation, as well as higher levels of transcription. So what does ubiquitination do to DOT1L? So the simple model was, well, it just recruits it, but it's not that simple because it really doesn't increase the binding the KD at all to nucleosomes. So what it does is stimulate the catalytic rate. And this was first shown by Rob McGinty, but this is our own data just showing Michaelis-Menten kinetics on an unmodified nucleosome versus a nucleosome with H2B monoubiquitination. So really a huge increase in the catalytic rate without affecting its binding. So in addition to being interested in what ubiquitin is doing, how does it bind, what does it have to do with stimulating methylation, another mystery about this particular enzyme was how it even got to its substrate lysine. So H3K79 is a lysine residue that's in the globular core of the nucleosome. And if you could see it over here in this blow up, H3K79 lies along the surface of the nucleosome. And there are now probably close to 100 structures of nucleosomes. It always lies on the surface, sort of buried in a valley and not accessible to DOT1. So although we've had structures for a long time of DOT1L, the human enzyme, as well as the yeast enzyme DOT1, it's never been possible to figure out how this enzyme, this side chain, gets into that active site. So shown in the active site here is S-adenosyl methionine, or SAM, so that lysine side chain needs to get up and access SAM, but yet something's got to change. There's got to be some sort of conformational change that would allow that side chain to get up into the active site. So to address these questions, a talented postdoc in the lab, Evan Morden, decided to tackle this by determining the structure of DOT1L bound to a nucleosome that's monoubiquitinated at histone H2B, lysine 120. So we can chemically ubiquitinate nucleosomes. He assembled complexes of the catalytic domain of DOT1L and the ubiquitinated nucleosome and determined the structure by cryo-EM. So this is the structure. People will know what this means, but this is a structure that's been determined at 3.9 angstroms resolution. So what's it tell us? So here's the structure of DOT1L bound to the nucleosome. And what you're looking at also is the map, the density map. So what we determine using cryo-EM, as well as with X-ray crystallography, is basically the outlines of the complex. So imagine putting shrink wrap around the complex. And then we interpret that map by docking in the structures. In this case, we actually had structures of all the pieces, and then we tweak them and look at the parts that have moved to fit the map. So that's what we're looking at here, the gray transparent region, and I'll give you a little tour of the complex now by dissolving away the map. So, sorry, that didn't dissolve yet. Okay, so here's the complex, here's DOT1L, here's ubiquitin, and now zooming in, we now see how it was that DOT1L is binding to ubiquitin. So remember I told you that DOT1L couldn't figure out how it bound ubiquitin, and it didn't contain any of the motifs or domains we knew about that recognize ubiquitin. Well, DOT1L just recognized it in a new way. So there's this appendage, if we look here on the right, this helix sticking out, and it forms, think of it like a little hand that's grasping this part of ubiquitin. There's no conformational change at all. It's this rigid element at the C-terminal end of the protein, and that's recognizing ubiquitin over here, so contacting these hydrophobic residues. So very nicely, Tom Muir's lab had actually mutagenized ubiquitin. So they had generated nucleosomes with ubiquitin containing a variety of point mutations to alanine substitutions to try to figure out which part of ubiquitin was responsible for stimulating DOT1L. And it's agreed actually perfectly with our observations. He had shown that mutating these two residues or substituting them with alanine were indeed important. So that was quite nice because we didn't have to do those experiments. I want to say going forward, with all the structures I've shown you, there are many mutations that we've made. I'm only going to show you a handful that we made to validate the structure, but there are many more than what I've put in my talk today. So we have this structure. We know how DOT1L binds to ubiquitin, and it's sitting on the nucleosome, but it was really clear right away that we were not quite where we wanted to be, because this was obviously an inactive conformation. And we knew that because of the distance between the substrate lysine. So this is the lysine over here, H3K79. This is S-adenosyl methionine. So in this case, it's modeled into the structure. We didn't see it, but we know exactly where it binds. This is the methyl group. And this methyl group is more than 20 angstroms away from the amino group and the lysine that it needs to methylate. So this clearly is not an active state, because the enzymes don't like pluck methyl groups off of SAM and throw it down onto the surface of the nucleosome. So there had to be another conformation that brought that side chain into the active site of the enzyme. So Evan thought really of a very clever way to try to trap this active complex. He had recalled that not in the case of DOT1L, but for this set family of methyltransferases. So these are lysine methyltransferases. MLL and Compass, which you'll hear about next, have a member of the set family of methyltransferases. And it had been discovered that if you replace the substrate lysine with a non-native amino acid, norleucine, that would bind incredibly tightly to the set domain amino acids, up to 3 orders of magnitude more tightly. So here's the comparison of the side chain. So here is the lysine over here, and basically norleucine is missing this NH3 plus group over here. So it's just the aliphatic side chain. So Evan generated nucleosomes that contain this norleucine residue in place of lysine 79. So he made histone H3, just histone H3, replaced the lysine with norleucine, assembled it into nucleosomes, added the ubiquitin and DOT1L, and that did the trick. So he really got, and it gave us a higher resolution structure. So this is actually, it was a 2.96 angstroms resolution. I know I should round to 3.0, but 2.96 sounds even more impressive. But what's really impressive is the quality of the map. So I told you earlier that we fit our structure to these maps. And there's really, I think you don't need to be a structural biologist to see the beautiful detail that we're able to resolve in these maps that really allowed Evan and Nick Hoffman, who played a leading role in this active complex, to build an accurate structure and even fit and discern conformational changes that occurred. So I'll show you now the active state nucleosome complex, and I'm going to compare this now to the poised state that I talked about. So here's the active state, and now we'll dissolve away the map. So this is the new state. This was the previous structure that I showed you. So this is the poised state, and we're morphing between the two states. And so what you could see is the ubiquitin serves as a pivot point. It's bound rigidly to that end of DOT1L, and it's twisting around. The flexible tail is allowing it to pivot around and adopt these two different positions. Now, another thing we noted in the active state, besides the fact that the enzyme is now down closer to the face of the nucleosome where it could get to H3K79, was the binding of this tail of histone H4. So this is a flexible tail of histone H4. And in the previous structure, it was actually disordered. If you look at various structures in the PDB, it's either disordered or it's lying across the DNA. It doesn't adopt any particular conformation. But here it was very specifically reaching up and binding to DOT1L and helping to position it on the surface of the nucleosome. And I'm going to get back to that tail in a minute because it plays multiple important roles. So the rest of the interactions here are mediated by the ubiquitin. So this ubiquitin recognition motif shown over here. And then this contact with the acidic patch. And I'll show you more about that on the next slide. So in addition to the contact with the ubiquitin, that is really the same in both of the complexes, There's also a contact with the surface of the nucleosome mediated by this arginine residue here. And again, we see that in both complexes. And the complex is actually pivoting around the two. It maintains this contact with the surface of the nucleosome and the ubiquitin, and those are serving as the anchor point about which it can adopt these different positions on the surface of the nucleosome. And this is really a critically important contact. So this is showing the activity of DOT1L. In light gray is on unmodified nucleosomes, and in dark gray showing you on ubiquitinated nucleosomes. And if we mutate this, if we do a charge reversal, we completely obliterate activity in both the ubiquitinated and unubiquitinated state. So this is really a critical contact. So this is really a common theme, actually, in enzymes that, or other protein complexes that interact with the nucleosome. So this just shows you a few previous examples here. There are actually even more, but the slide would be too cluttered, where proteins that interact with the face of the nucleosome have some kind of arginine anchor, as that Song Tan came up with that phrase, that anchor these complexes to this acidic patch. So this acidic patch is a conserved cluster of acidic residues between histones H2A and H2B that mediate these important electrostatic interactions. It's interesting that so many things go for the acidic patch. The patch is important in actually making a more condensed chromatin because the charged tails of neighboring nucleosomes can actually bind to those patches. So perhaps in these cases, the targeting that acidic patch perhaps helps to maintain the more open state of chromatin, which you need for transcription. Now, I told you that one of the mysteries was how DOT1L even accessed its substrate lysine. And this, in this slide here, shows the conformation of the lysine in the poised state nucleosome, which is what you see in pretty much every other structure. So this was really incredibly surprising because what we found is that the conformation or the position of that side chain changes dramatically in the active state. So it swings up by 90 degrees and now is pointing directly off the surface of the nucleosome and is now in a perfect position to initiate the reaction, the methylation reaction. So in this position, it's now about 3 angstroms away from the methyl group of S-adenosyl methionine. So exactly where it needs to be for the chemistry, the methylation reaction to occur. And I'll show you that conformational change here on the right. So if you watch, we're going to morph to the active state and watch what happens on the surface of the nucleosome here. You see that comes all the way up into the active site of DOT1L, where it can initiate the methylation reaction. So this was an incredibly surprising result because in all of the structures of the nucleosomes, that globular core is always the same. There's virtually no change when you're looking at an intact nucleosome. So this was incredibly surprising that it wasn't really DOT1L that underwent conformational changes. There are a couple of little movement of loops, but the globular core of the nucleosome itself. And I'll show you another picture here. So we're looking at this from the side. So this is showing the helix in histone H3. So the dotted lines show you the hydrogen bonds. And what you're going to see is in order for that conformational change to occur, we're going to rip apart those hydrogen bonds. So they're pulling away. So you could see in the morph here, they're lengthening those distance and breaking those hydrogen bonds. This is energetically very costly. You know, that's a favorable conformation and you're pulling it away and really paying a price for breaking all of those hydrogen bonds. And this shows you again the distance to SAM. So another aspect I told you that we saw in the structure was the tail of histone H4 coming up from the surface of the nucleosome and binding in this groove in DOT1L, shown over here. So the tail of histone H4 So the density is illustrated over here. And there's this cluster of residues. There's an arginine and a histidine shown over here and another arginine. And these are mediating contacts with both DOT1L as well as with histone H3. And so this actually explains an observation from quite a while ago that was first made in yeast and then with human DOT1L, that tail of histone H4 was incredibly important for methylation. So what's shown here is a Western blot plotting against the H3K79 monodi and trimethylation. So this is the wild type nucleosome, and this is showing a series of mutations in the nucleosome. So different side chain substitutions and charge reversals. And if you substitute either of these arginine residues, you knock out methylation here. So this had clearly shown the importance of histone H4 in somehow promoting methylation by DOT1L. And these interactions here explain why that tail of histone H4 is so important. Now, it turns out that that tail of histone H4 is not simply helping to position DOT1L in the active conformation, meaning in its position lower down onto the surface of the nucleosome. It's actually helping in that really critical conformational change that needs to occur to get that lysine up into the active site. So I'm going to show you the morph again that I'd shown you earlier, where we're going to rip apart the helix and swing the side chain up in the active site. But what you'll see, it's actually the histone H4 that's helping that. So I'm going to morph again. So there's lysine 79. And we're going to morph now into the conformation and the active complex. Okay, so I pointed out before that ripping apart or increasing the distance here and disrupting these hydrogen bonds is something that is energetically costly. Histone H4 helps by coming in and now forming a new set of contacts. So that arginine side chain is forming hydrogen bonds with multiple carbonyl oxygens over here. So we pay an energetic cost for tearing apart those hydrogen bonds, but we gain some energy by the tail of histone H4 coming in and forming a new set of interactions. So it's helping to pay energetically for distorting histone H3, sorry. So here's that arginine again, and of course there are multiple other interactions between DOT1L. There's a phenylalanine that actually props up the other side of histone H3 that has been pulled up off the surface of the nucleosome, and then it's surrounded by other hydrophobic side chains. If we mutate any of these, we really kill activity of DOT1L. So those hydrophobic side chains form this tunnel around that lysine side chain, and you'll really be able to see it here. So what I'm going to do is show you a movie where we're going to look from underneath the nucleosome, as if we could really do that. And what you'll see is this hydrophobic tunnel. So we look from the bottom there, and this is the surface depiction now of DOT1L. So you can really see that narrow tunnel that lysine side chain is inserted into. So this helped answer another lingering question about DOT1L, and that is, how does it catalyze the reaction? So the reaction, that lysine side chain is normally charged, but it needs to be deprotonated in order to initiate this attack on the methyl group. And it was a lingering question as to what was it in the enzyme active site that deprotonated the lysine. No one could ever identify an active site residue. And with our structure here now, we also see no active site residue that's deprotonating it. And so what must be happening is actually by basically shoving that lysine into this very hydrophobic environment, it lowers the pKa of the lysine. This is a known phenomenon. Lysines in hydrophobic environments have lower pKa, meaning they're less likely to be protonated. And so that environment alone sufficiently lowers the pKa to allow this enzymatic reaction to occur. Okay, so to summarize this part of my talk, so I showed you all the mechanics of how DOT1L recognizes ubiquitin, how that restricts its movement on the surface of the nucleosome, and that restriction of movement can account for the increased catalytic rate. It's basically kind of limiting the search of DOT1L for its substrate lysine from diffusing around to basically just being able to pivot between these two orientations. So that's like this robin over here. Excuse me. So like this robin over here that's sitting poised, looking up alert for a worm, and imagine the ubiquitin is holding its tail over here. And then when it sees a worm, it can pivot down onto the ground and pull that worm up out of the ground, just the way DOT1L is pulling K79, lysine 79, off the surface of histone H3. And then this tail of histone H4 is assisting in that conformational change. So this is you could think of this as a case of substrate-assisted catalysis, where the other part of the nucleosome is playing a critical role in aiding in this process. So the finding that the nucleosome undergoes a conformational change has important implications, because we think of recognizing the core of the nucleosome as basically, there's the rock, that's what the nucleosome looks like. And so anything that recognizes it is simply adapting to this very stable structure. So if DOT1L can distort the surface of the nucleosome to remodel those globular histones, there are likely other proteins that do the same. In particular, we actually don't know of any of the reader domains that recognize the H3, K79, any of the methylated states. So it's possible that when we find them, and we hope to find them, that they too will introduce some sort of distortion. And this is actually in keeping with recent work from Geeta Narlikar and John Gross at UCSF, which have found that particularly when nucleosome remodeling enzymes are acting on the nucleosome, that core is actually more plastic than we had previously appreciated. These experiments were by FRET and NMR. So it's very cool to actually see an example right here in our structures. Okay, so in the next part of my talk, I want to tell you what we've learned about crosstalk between ubiquitination of H2B and that other methyl mark, H3K4. So this is a mark here now in the flexible tail of histone H3, not in the globular core. So histone H3K4 methylation is found near the transcription start sites. It's also enriched in enhancers in human cells. And again, levels of that methylation are associated with correlating with levels of transcription. The higher the levels of mRNA transcription, the higher the levels of methylation. And this again shows a result of, from 2002, showing that if you knock out any of the machinery responsible for ubiquitinating histone H2B, so RAD6 is the E2, you knock out or knock down methylation of H3K4. So I already referred to this earlier, but the role of H3K4 in activating transcription is several-fold. The SAGA complex contains a histone acetyl transferase subcomplex that it contains GCN5 along with other proteins that recognize H3K4, and that actually stimulates SAGA to hyperacetylate histone H3. The chromatin remodeler, CHD1, has a domain that recognizes it, and there's the methyl mark also helps to stimulate assembly of the transcription pre-initiation complex. So those are some of the roles of histone H3K4 methylation. Now, the enzyme complex that modifies this is way more complicated than H3K79. So as first nicely elucidated by Ali Shalati in yeast, it's this large complex called COMPASS. The methylation is mediated by CET1, so it contains the C-terminal catalytic domain, a CET methyltransferase, and this important region, the NCET domain, but all these other proteins here are associated with it as well. And it's been shown from work from a number of labs that this constitutes the minimal complex. So 5 proteins and these fragments. So this part of set 1, these 3 proteins here, and then these 2 proteins here together are the minimal complex that are required to methylate H3K4, but also to be stimulated by the presence of ubiquitinated H2B on the nucleosome. And the counterparts of these proteins in the human MLL core complex are listed over here. So the structure of the yeast complex was determined by Ali Shalati and Yorgos Skiniotis a couple of years ago, and that's illustrated over here. So this is the minimal complex that's required for H2B ubiquitination. And I should say there's also a structure of the K-lactis, so another yeast complex from Ning Zhang's lab. So Evan again decided to tackle this as well, and as long as he had nucleosomes with ubiquitin, and this time he substituted the lysine 4 and histone H3 again with norleucine, so that non-native amino acid lacking the charge NH3 group, and determined the structure by cryo-EM. And this structure was determined to a resolution of 3.4 angstroms. So COMPASS is really a very different beast. It's this. So here's this large complex, and we're seeing 2 views here of COMPASS. So this really spans the whole nucleosome surface. We're looking at a side view over here, and this is a view from the top where you really can see that these proteins virtually cover the surface of the nucleosome. So I'll show you a couple of views. So here again, this is our density map, and we'll dissolve it away to look at the structure of COMPASS bound to the surface of the nucleosome. So you really see it crosses it completely here. What I'm pointing out here is the catalytic domain. So this is set 1. This is the catalytic domain, S-adenosyl methionine, and here's the tail of histone H3 that's bound to the catalytic domain. So interestingly, the set1 methyltransferase actually is targeting the copy of histone H3 not on the same side of the nucleosome, which is what DOT1 does, but on the opposite face of the nucleosome. So we don't actually see the connection between this tail of histone H3, the N-terminus, and the globular part of H3. It's a residue 38 over here. So this is one copy, and then the other copy is over here. So we had to compare what is this attached to? So this is 88 angstroms away from the copy of H3 that sits on the top of the nucleosome on the same side as COMPASS here, and this is the copy that sits below, and just based on distance alone, this can't reach over here. And so it's clearly this copy of histone H3 that's being modified. And so we think of this as transhistone recognition, ubiquitin on one face and modifying the copy of H3 that's on the other face. Now, in the case of DOT1L, DOT1 didn't change conformation basically at all, but was just pivoting, and then we had that conformational change in the nucleosome for the substrate to get up in the active site. Here we have the opposite. We have a large number of conformational changes where COMPASS adapts itself to the nucleosome. And I'm going to morph and show you those changes here. So we're looking here at the structure of yeast COMPASS from Yorgos Skiniotis' lab. And what I'm going to show you is a morph between this apo complex and the conformation that we see bound to the nucleosome. Oh, right. Before I do the morph, so this is where the ubiquitin is in the final complex, and now we'll see the morph around that. Yes, okay. So look at how that complex is flexing over the surface of the nucleosome and adapting itself to the surface over there. Okay, so this is the nucleosome bound structure of COMPASS. So it's spanning the nucleosome. I showed you how it's binding the tail from the copy on the other side, and it's also contacting the DNA. It's another difference. DOT1 does not contact the DNA at all, at least not the piece that we have in our structure, but compass on either side, actually these two proteins are mediating contacts with the DNA, and substitutions in those residues knock down methylation when we look in vivo and in vitro experiments done by other labs. A critical conformational change also occurs in this little helix over here. So it had been shown by Bob Rader's lab that there's this N-terminal segment in set 1. They call it the N-set domain, or the RXR helix. And it was found that this is essential for activity on nucleosomes and is essential for this complex to be activated by histone H2B. And this is really sort of a linchpin here. You could see this view over here. So this is the helix, and we're now going to we're looking down the helix over here. You can see how it's wedged in between the ubiquitin that's above and the surface of the nucleosome below. So what does the helix do? Why is this important? So part of this helix is, again, contacting the acidic patch of the nucleosome. So here are those acidic residues again, the glutamic and aspartic acid residues. And here's a cluster of arginine residues. So again, a cluster of arginines interacting with the acidic patch. So that's one set of contacts. And then the other end of this helix contains a set of hydrophobic residues. And these hydrophobic residues are interacting, again, with that same hydrophobic patch that DOT1L was contacting before. Okay, so this is really a linchpin, contacting the ubiquitin and contacting the surface of the nucleosome. So gluing that complex together. So there are really amazing conformational changes that occur in order to create that binding helix. I'm going to show you a morph here between the apocomplex and the complex bound to the nucleosome. And you watch how that strand undergoes a conformational change, and this helix, which isn't present at all in the previous complex, now forms. So this changes conformation actually helps to nucleate formation of that helix there. There are more conformational changes. So this protein over here, SWD1, also undergoes a conformational change to form this hydrophobic patch that you'll see that is then critically important for contacting actually another portion of ubiquitin. It's called the I44 patch. Yeah, there we go. Here's ubiquitin here. So this is now forming a second contact surface with the ubiquitin. So how is ubiquitin activating this complex? So it's clear that the contacts with the ubiquitin help to order this helix. This is this critical linchpin that is contacting the surface of the nucleosome, and it needs the help from these contacts with the ubiquitin to help to nucleate formation of that helix. Although I should say, in full disclosure, actually part of this helix is already ordered even in the absence of ubiquitin, at least as been shown by, oh, that's actually a mistake. It's not S-Pombi. It's actually the structure of the K-lactis compass that was determined in Ning Zhang's lab. But still, ubiquitin this is a partially disordered helix. So ubiquitin is clearly playing an important role in completing the ordering of that helix and forming that critical contact with the surface of the nucleosome. Okay, so what are the common themes here in crosstalk between ubiquitin and methylation between these very different enzyme complexes? So they both have these arginines that bind to the nucleosome acidic patch. In the case of DOT1, it's the ubiquitin that's helping to restrict the conformational search of the enzyme, whereas here that may be true, but the ubiquitin is helping to favor a conformational change in this helix, which in turn is important for interactions between the complex and the nucleosome. In the case of DOT1L, we also have this very fascinating example of substrate-assisted catalysis. So the tail of another histone assisting in this conformational change and helping DOT1L to reach its substrate. And it could be that the ubiquitin, in addition to the contacts with the H4 tail, that the interactions with ubiquitin also help to pay for this energetic penalty for the distortion of H3K79. And again, here also ubiquitin clearly also stabilizes the compass conformation that is competent for binding and catalysis. So, so I've shown you the mechanistic insights that we've gained from the structural studies of these two very different enzymes, both of which are stimulated by ubiquitin, and where have ongoing efforts to actually use this information and exploiting it for drug discovery. So these structures give us insights into the little crevices and the interactions among the proteins that are very important for stimulating the enzymes and for their activities. And so that provides avenues to think of ways to design new small molecule inhibitors that specifically inhibit this particular class of methyltransferases, as opposed to every single methyltransferase of every type in the cell, all of which use S-adenosyl methionine. And most drugs that have or small molecule inhibitors, by and large, target that active site, rather than these protein-protein interactions that are visible from these structures. And so these are, we're ongoing efforts using our structure of DOT1L, compass, and actually unpublished structure of MLL1, so the human H3K4 methyltransferase, as well as the SAGA dub module. I really didn't talk about this here, but the human homolog, this is yeast, but the human homolog is also the catalytic subunit, is overexpressed in a variety of cancers and represents an attractive drug target. And these are our collaborators in those ongoing efforts. So I want to give the credit to the people who actually did the work. So this is much of the work is from a phenomenally talented postdoc, Evan Warden. If anyone out there is listening that has an open position, Evan is awesome. And also Nick Hoffman, who worked on the high resolution structure of DOT1L. And in these projects, also student Chad Hicks, shown over here, and my longtime technician, Shangbin Zhang, made very important contributions. And this is the lab. It makes me very wistful because we're not allowed to do things like be close together like this. And finally, I just want to thank the sources of funding for this research. And thank you all for your attention, and I'd be happy to answer questions.
Thanks very much, Cynthia, for a fantastic talk. So everyone should put in questions into the Q&A box. So far, we have one question from June Chi. Do you think the inhibitor binding to DOT-L1 will interrupt this interaction or even the ubiquitination? So I think this is a question related to the last slide you were talking about.
So interrupting the ubiquitination?
Interrupt the interaction or even ubiquitination, or maybe he meant methylation.
Yeah, so that's the hope, is that we can, so we're looking for, small molecules that can interrupt the critical interactions that are required for that conformational change, or for stabilization of that active complex on the surface of the nucleosome. There's one inhibitor that's in clinical trials, pinametastat, that targets the active site. I'm not sure that's been very effective thus far, but the hope is that we can target some of those other interactions that are critically important for activity.
So I was curious about the different conformations of ubiquitin on the histone octamers. So there's obviously multiple different structures that have ubiquitin there. But what are the dynamics of ubiquitin? And obviously you're getting a preferred orientation when you have DOT1 or other proteins bound. But I was just wondering what the dynamics are, and is there a preferred orientation or something similar to that you can see across the different ubiquitinated histones?
Yeah, so I think I may even have a slide here. Oh, isn't that amazing?
That wasn't planned, by the way.
I know, but thank you for asking that question. Yeah, so this is a slide that superimposes the three different, actually I didn't put MLL, but There really isn't. You know, as you know, ubiquitin has that flexible tail, and just comparing even DOT1L and COMPASS, it's sort of in about the same position, but rotated a bit. In the SAGA deubiquitinating module, it's lying flat. You know, we have seen in structures, so as the cryo-EM people will know, so often we make these complexes, put them on the grid, and then they fall apart. And so we do have lower resolution maps of nucleosome that just have ubiquitin on it. But so it's interesting you could see the ubiquitin, because that's telling you that at least in a subset of cases, it is lying there with a preferred location, although we can't tell the relative orientation on the nucleosome. But I actually doubt that that's playing a role, and that's actually something that's being recognized.
So it went by a little bit fast. I didn't really notice, but it sort of looked like, is the I-44 face used to engage the different components, or not so much?
Yeah, so the I-44, yeah, I removed some of the very ubiquitin-centric slides, so apologies for glossing over that. So in the case of DOT1L, it's not recognized at all. And actually, Tom Muir's lab had shown that you can attach a ubiquitin with I-44 change to alanine and to a nucleosome, and it activates DOT1L just fine. Whereas it does not activate COMPASS. So COMPASS is contacting both. It makes much more extensive contacts. It has one of the proteins that undergoes a conformational change is recognizing I-44, and another one, that very critical RXR helix, is recognizing that other patch, you know, I-36, you know, 73.
Okay. So the next question is from Steve Blacklow. Does the substitution of lysine with norleucine simply favor the active conformation because of the energy required to deprotonate the lysine amine?
That's our guess. That's our guess, because you're removing, right? It costs a lot of energy to insert a charge residue into a hydrophobic environment. My colleague Bertrand Garcia Moreno studies this in great detail. And so there's actually a pretty significant penalty in binding. So the norleucine presumably gets rid of that penalty. And actually methionine works as well. And, you know, norleucine works the best, but even just substituting a methionine side chain in there also increases binding. And so you probably is just favoring that conformation, which obviously has to occur stochastically with the lysine residue, and you're just tilting the equilibrium, is our thought.
Thanks. So the next question is from El Shama. I didn't get what made H4 tail change its position and bind to H3 and form an energetic hydrogen bonds.
Yeah, so that tail of histone H3 is very floppy. And it just, you know, there are about something on the order of 30 disordered residues there. So COMPASS basically sits there. And as far as we know, that tail just, you know, you've basically, it essentially now has a high local concentration of its substrate, which is the tail of histone H3, and that makes its way into the site. So it's also reducing conformational search in some sense, but in this case, it's the tail that flops around and then lands in the SET1 active site.
So the next question is from Randy King. Once the methyl marks are created, does the presence of ubiquitin affect the rate of demethylation? Furthermore, does methylation influence the rate of deubiquitination?
Those are very interesting questions that we are pursuing. So, you know, it's relatively recently now that you really have available some of these modified nucleosomes. Actually, the company EpiCypher has been set up. I don't make any money from EpiCypher, but you don't need a collaboration with a chemist. You can actually obtain these substrates. And so we're looking at exactly that question. I should say that for K79, there is no known demethylase. That's really it's known for H3K4. We don't know of a demethylase for H3K79. We don't know of readers for H3K79. So those are incredibly interesting questions to pursue.
Next question is from Mike Eck. Curious, and this is sort of related to Steve Blacklow's question. Curious where the norleucine is located on the DOT1 complex and how it is stabilizing the active conformation of the complex.
I mean, we presume that it stabilizes the active conformation by simply shifting the equilibrium. So it basically should bind more tightly. And actually, I don't have the gels here, but Evan has done gel shift experiments, and you do see tighter binding of DOT1L to nucleosomes containing the norleucine versus the lysine. So it clearly binds more tightly and is just tipping the balance. One other thing, if maybe you're thinking, is, and I didn't show this image either, but the norleucine is not affecting the conformational change because a subset of our particles in the cryo-EM structure determination only have DOT1L bound to one face of the nucleosome. And in those particles, there's no conformational change in histone H3. So that's sort of the control for what does the norleucine do? You know, nothing unless DOT1L is there.
The next question is from Lori Glimcher. In your search for drugs, are you docking to specific sites on complex structures, or have you designed or devised a more general cellular screen?
So yes to the first question. We have not done a cellular screen. Right now, we're just looking in vitro, although obviously we need to then move to a cellular screen. The challenge I shouldn't say challenge, but it just makes it harder to screen DOT1L because for reasons that I still don't completely understand, it is only active on nucleosomes. It is not active on peptides. So all those screens, you actually need intact nucleosomes with ubiquitin on it, because without ubiquitin, the signal is pretty terrible.
So the next question is from Eric Fisher. Following up on the idea of specificity, can you move the ubiquitin attachment point on the histone by 1 or 2 amino acids and retain recruitment activity? If so, any other factors that would aid selective recruitment of one over the other histone modifying complexes?
So I know that, so this has been done artificially, and I apologize, I don't remember the paper, but someone has done it, I believe it was for DOT1L, but I'm not 100% sure, where they basically attach the ubiquitin to the very C terminus of H2B, and it was still able to stimulate. So it doesn't look as if it's a completely strict requirement, but I don't remember the details of those experiments. It's not work that we've done. As to whether there are other molecules that could cause it to bind there and substitute for the ubiquitin, which I think what you were asking is a very interesting question, and I don't know, but that would be incredibly interesting.
So I think this possibly is the last question from Brenda Schulman. In the DOT1 complex, does the H2B linked ubiquitin via its I44 patch recruit something else into the complex? Sort of like NET8 linked to COL1 uses both the I36 and I44 patch.
Yeah, so we don't know, and that's an interesting question. So DOT1L actually forms these higher order complexes with these other partner proteins, and then they're implicated in MLL rearranged leukemia. And that's not, there's a little bit of structural information on them, but there's really not a lot of information on the larger complexes. So I don't actually know, you know, there's no experimental evidence addressing this. In yeast, there aren't these higher order complexes, so you can't do those experiments in yeast to look at it, but it's a really interesting question, and it wouldn't surprise me if there was, but we don't know that yet.
Well, it looks like perhaps no more questions. So I think we're right on time. Thank you very much, Cynthia, for a fantastic talk. And
thank you for the invitation.
Hopefully we'll see you sometime soon.
Yeah. Hello to all the people out there that I know. I'm sorry this is virtually and not in person, but hope to see you all soon.